Thank you very much, Scott. Oops. For disclosure, I want to point out that I do have an ownership interest in a company. And I'm going to talk about two off-label drug applications during this talk. The talk is going to be divided to address these issues. I want to talk first of all about basic mechanisms to give you an idea about why we're focusing on this particular pathway, why ODC. It's really the rationale for the pathway approach. I'm going to talk about some clinical trials at the beginning that have validated this pathway, we think. And then I want to spend the last part of the talk really focusing on the ODC genotype, both in terms of disease prognosis, but especially in terms of response prediction. And for response prediction, I'm going to focus on both the benefit aspects of this as well as patient toxicities. In terms of the basic mechanisms, this is a little review from Jeff Green several years ago that outlined a number of pathways involved in colon carcinogenesis. So I want you to think about this, at least from our perspective. We started from this thinking about colon carcinogenesis. Generally, there are clearly a number of pathways, number of genes that are involved. Our focus happened to be on the APC gene here in the Wnt signaling pathway. And we used a number of technologies, microarray and other approaches to try to understand APC-dependent genes, but genes that were regulated by inflammation. And I'll just bring your focus down here. One of those genes happens to be the MYC oncogene. In the last 15 years, we and other people have painstakingly connected APC and MYC to a number of genes. One of the pathways that has attracted our attention especially is this polyamine pathway, an amino acid decarboxylation pathway. And from a little review I wrote earlier this year, just pointing out that MYC regulates the expression of a number of genes, the first enzyme in this pathway, ornithine decarboxylase. But it's not just ornithine decarboxylase. MYC actually regulates the expression of a number of genes involved in the metabolism, a protein that's post-translationally modified by these polyamines that have oncogenic potential. So this is the pathway that we've followed. We've targeted this pathway pharmaceutically using a number of agents. Here I would just remind you, we're talking about a number of steps in this pathway. Biosynthesis, ornithine decarboxylase is just one of the genes. There are clearly a number of other processes, catabolism, uptake, efflux mechanisms. In our experimental studies, we focused on one particular agent, difluoromethylornithine, which is an inhibitor of ornithine decarboxylase, in combination with several nonsteroidal anti-inflammatory drugs. And I just draw your attention to the fact that most of you would pass the test if I asked you what do nonsteroidal anti-inflammatory drugs do, and you'd say they inhibit cyclooxygenases, and that's true. But these nonsteroidal anti-inflammatory drugs have a number of non-cyclooxygenase-dependent mechanisms, one of which is a mechanism that we've identified, which is actually they promote polyamine efflux through a very specific mechanism that I'm going to talk to you about later. So we've targeted this pathway using these two agents, although there are clearly other points of intervention in this pathway. Now, to set the basis for why the polyamines, why ODC, I want to start with a study actually that was led by my colleague Frank Maskins, who's here in the audience, a study that we did, a clinical study in people with prior colon polyps. That was done from 1997 to 2007. It had 375 patients. 267 of these patients went to final randomization or final colonoscopy, and it was a placebo control to the combination versus the combination of this drug difluoromethylornithine and sulindac. The primary endpoint was whether or not there were adenomas at 3 years, although we looked at a number of markers, including genetics, and that's what I'm going to focus on after we talk about this trial. But in terms of validation, Peter Greenwald was very generous yesterday in noting this trial was extremely effective. There was a 70% reduction in total adenomas in response to this particular treatment. More importantly, we think there was a very dramatic reduction in advanced and multiple adenomas. The first important thing is this validates this particular intervention. I want to draw your attention, though, to this 30% number in the treatment group that didn't respond to the treatment, or at least didn't have total reduction, because it's this number that we're really going to focus on in terms of some of our failure analysis in just a second. Another point about the validation, though, is if I was you in the audience, I would be skeptical and I'd say, well, great, so you've used these drugs. If NSAIDs have non-cyclooxygenase effects, maybe there are other effects that are caused by these drugs. And in terms of looking at genetics, so for predictive markers, you should be thinking about something else. This is from a paper that one of my colleagues, Patty Thompson, recently published in gastroenterology, that is an analysis of the biomarkers in the tissues documenting that the targets of these drugs, these are the polyamines. What you're looking at here are the number of adenomas. So this is basically an assessment of those patients, placebo versus treatment, that responded, whether or not they had low polyamine contents, so that would be this group and this group, or high polyamine contents, little difference between placebo and treatment. So this top panel is data indicating in this population that baseline biomarkers in this pathway predicted response to the treatment. The bottom panels are a series of data that just are for the placebo arms and the treatment arms. Same thing here, placebo arm, treatment arm. These are PGE2 levels, and they're at baseline 12 months and 36 months. And what this shows on the bottom panels is here that the treatment intervention causes especially a statistically significant decrease in the tissue polyamine contents. So I show these data to emphasize the point that we have clinical data that intervening in this pathway is a very potent way to suppress metachronous adenoma formation in people with prior colon adenomas. It also is a treatment that targets the pathway that we think we're targeting. So this is the rationale for why, from here on in, I have a very laser focused on polyamine metabolism. Okay, ornithine decarboxylase is one gene in the polyamine pathway. But as I've emphasized, there are a lot of other genes in the pathway, and in terms of thinking about genetic variability as a potential predictor for responses, we're certainly looking at all of the genes in this pathway. But I just wanted to spend a couple of minutes emphasizing the point that if we focus on one gene in the pathway, ornithine decarboxylase, there's a lot of genetic variability in this one gene. What's shown here, the NCBI model for the ornithine decarboxylase gene, the blue upticks are the exonic sequences. The gold and yellow upticks are regions or are points of putative genetic variability or single nucleotide polymorphisms. The second line is a blow-up of this first intronic region, and this third line is a blow-up of one particular region in this intronic region. Now, we have sequenced this whole gene in our clinical trials to determine actual allele frequencies of these particular polymorphisms, and they're shown here. We have established through both our own sequencing data and also publicly available databases haplotypes that account for the majority of us in terms of this particular genotype, and they're shown here. And I just wanted to comment that we are looking at the functionality and the associations of these, especially these haplotypes, because we think these are going to be the most significant for predicting both prognosis, disease prognosis, and prediction. With that said, from here on in, you're going to hear me talking about one polymorphism, and that's this polymorphism here that's in the red box. It's the second SNP that's noted right here, and if you look at the sequence underneath, some of you may recognize consensus EBOX transcription factor binding sites here that are underlined and here that are flanked by this particular polymorphic site. And I understand that some of you have the perspective that polymorphisms, we don't really care about their functionality, we care about their associations. I personally care a lot about the functionality, and so one of the things that we've done is we've spent a lot of time, looking at the functionality of these SNPs. This is one piece of data for functionality of this one SNP at 316. And what it shows is an analysis called CHIP analysis for chromatin immunoprecipitation analysis. Basically, you treat cells, you cross-link DNA and protein in intact cells or tissues. You then isolate the DNA or the chromatin, you fractionate it. You can use antibodies to pull down protein sequences, dissociate the antibodies, and identify where the proteins are in fact binding in the DNA. And what we've used a couple of different colon cancer-derived cell lines here that are either homozygous at this particular 316 locus or heterozygous at this 316 locus. These are just Western blots, and this is work that Alex Bobbs and Ning Q in my lab did, documenting that these cells express all of these EBOX transcription factors. But as shown on the bottom in the CHIP analysis, you only get binding of these proteins to cells that have an A allele, not a G allele. The other thing that's remarkable about this data is it shows that both the transcriptional activators, MYC, and the repressors, the MAD family of transcriptional repressors, bind to the A allele, do not bind to the G allele. In working with a number of colleagues, we've associated this polymorphism with clinical outcomes. The data on the left and the bottom is work from Elena Martinez, my colleague at the University of Arizona. The work on the right in the bottom is from Jason Zell and colleagues at the University of California, Irvine. This data comes from patients with prior colon adenomas and gives an odds ratio for the risk of developing a new adenoma within 3 years after a qualifying colonoscopy. And what you can see is that individuals who are homozygous A for this particular allele have about a 50% reduction in their risk of developing an adenoma after 3 years. And if you look at this little cartoon on the top, what we think is happening in the normal colonic mucosa, where in our animal studies and human studies we've shown expression predominantly of repressors, the repressors are binding to this A allele, thereby suppressing this pathway, leading to a reduction in risk. The surprising finding came several years later, actually about a year ago. We published work with Jason Zell, who looked at about 500 patients in the Southern California Cancer Registry, patients with colon cancer. These are colon cancer survivors. Note the long follow-up times on these patients, where he then associated the ODC allele with survival of colon cancer patients. Here, the ODC A-allele is a protective factor. Here, the ODC A-allele is a highly statistically significant risk factor. And what we think is going on, based on our mechanism studies, is that in colon neoplasia, now think about adenomas, activation, or there's loss of functional APC, activation of MYC. MYC is now going to be expressed in these tumors. It now becomes a promoter of this pathway. In either case, these data argue that this is an important pathway for colon carcinogenesis. Okay, thinking now about prediction of response to treatment. As part of our trial, the Elena Martinez trial in 2003, and that's referred to in this line up here, but there are now 2 independent studies that have evaluated the frequency of this SNP in association with aspirin usage in In our trial, it actually was a trial for another another cancer prevention intervention, wheat bran fiber. These lower two studies, Elizabeth Berry's study and the Huebner study, were for prospective randomized studies of aspirin intervention. And you can see the numbers here of patients in the trials, the numbers genotyped, over 2,000 patients genotyped, similar ODC A-allele frequencies, the overall frequency of the genotype in the populations. These are all three predominantly Caucasian populations. And the remarkable finding that were noted in all three studies is that there appears to be a dramatic association of this A allele with a decreased risk of developing adenomas in people who are taking aspirin, either reporting taking aspirin, as it was done in our study, or actually a prospective randomized study of aspirin. So it gets back to my first point again in terms of this is a test for you all. What does ODC have to do with nonsteroidal anti-inflammatory drugs? To remind you, we actually started, again, from basic principles. We looked at microarray analysis of NSAID-inducible genes. We discovered a polyamine catabolic gene called the spermidine acetyltransferase, or SAT1, is a target of a number of nonsteroidal anti-inflammatory drugs. Sulindac appears to work to activate this through a transcriptional mechanism involving PPAR gamma, but a number of other NSAIDs, including aspirin and Celebrex, also activate this gene, and they do so by unique transcriptional mechanisms. Natasha Gnatenko in my lab was the person who did most of the animal studies. We validated this result in animals, and Takashi Uemura is currently working on this export phenomenon that we think is relevant to some of the clinical studies that I'll talk about at the very end. Now, we validated this idea about NSAIDs actually working through the polyamine pathway. One of the ways as a biochemist that I thought about this is that when we do these studies in cell culture, we oftentimes do rescue experiments. So if we use an enzyme inhibitor, we block a pathway, we restore the pathway, we can ask whether or not we affect the phenotypes. This is a rescue experiment we did in a mouse model, the Min mouse model. So this is an APC mutant mouse model, develops intestinal carcinogenesis. These mice in our hands get about 35 tumors per mouse. If you treat them with high doses of the nonsteroidal anti-inflammatory drug sulindac, you can get a very potent reduction in tumor number. If we feed these mice 1% putrescine in the drinking water, this corresponds to the amount of putrescine that you all would consume if you drank 2 glasses of grapefruit juice every day. What you can see is that there is little effect by itself in terms of total tumor number, but in terms of the sulindac effect, there is a very dramatic reduction in the ability of sulindac to reduce tumor number in mice that are simply fed a relatively low amount of this polyamine putrescine. This is the same kind of a rescue response that we got when we did this experiment in cell culture. And this just documents that the animals that were consuming putrescine had elevated putrescine levels, they had elevated spermidine levels. There was also an associated change in the adenomas that developed with a significant increase in the animals consuming putrescine in terms of their adenoma grade. Think back to our clinical trial in terms of the advanced adenomas and the multiple adenomas. We extended this finding, have extended this finding now into humans using the clinical trial that I talked to you earlier about. This is work from Kavitha. Actually, I wanted to, first of all, just tell you about how we have quantitated polyamines in human diet. This is work actually from Cheryl Rock's lab. Christina Zimas-Morris was the graduate student who worked on this. We actually converted information in a food frequency questionnaire to data relating to polyamine contents, because many of the foods that these polyamines are found in all kinds of plants and animals. It's known that certain food types have amounts of various polyamines. I'm only showing the putrescine data here for some amounts. These are amounts that are in diets from patients that were in our trial. We computed those foods that contributed the most of these amines. So you can see, for instance, citric juices, which use these polyamines in ripening, but also processed foods, meats, cheeses, and so on, appear as high components of these amine contributors. And this is now work from Kavitha Raj and Jason Zell at UCI that have associated the polyamine contents. So if I could just go back to remind you, we've done the dietary questionnaire. We have measured rectal tissue polyamine contents. We've used that dietary questionnaire data to also then associate tumor responses. And that's what's shown here, this top table from our clinical trial without any dietary communication with the patients, simply asking them what they ate, are divided into the lowest 3/4 or the highest quartile of polyamine consumption. And you can see that the people who consumed the highest polyamine levels had statistically significantly elevated levels of polyamines in their rectal mucosa, just like in our mouse studies. And more importantly, down here, if you look at the response of these patients to the treatment, for 75% of patients, the lowest quartile, all had highly statistically significant responses to all of the adenoma endpoints. But in the patients who were in this highest quartile, think about the overall clinical data. We had a 30% failure rate. Those individuals had essentially no benefit. So it begins to be a way of selecting out those patients that are failing from the treatment. Now, the last part of the talk, I want to focus on toxicities. We've done a broad-scale toxicity assessment in all the patients in this trial. We focus especially on hearing loss and ototoxicity because this is known to be associated with this drug difluoromethyl ornithine. I would point out to you that in our placebo group, the definition of our ototoxicity was greater than a 15 decibel hearing loss in 2 consecutive frequencies. That occurs in about 10% of us simply as we get older. In the treatment group, there was about an additional 10% of patients that appeared to be associating or having this ototoxicity. This was a statistically significant effect, even though it was not a clinically significant effect. It was statistically significant because we did quantitative audiology. The patients, in fact, did not report this as a hearing loss because this occurs predominantly outside of the human hearing range. Doesn't mean it's not important, and it clearly is potentially an indicator of future problems. So it's something that we're very concerned about. We've looked at a number of other toxicities. We've published about this cardiovascular toxicity that we think is associated, and other people think are associated with the use of nonsteroidal anti-inflammatory drugs, and I'm going to come to this at the very end. But in terms of this hearing loss, and in thinking about the ODC genotype, again, our focus is what's the pathway, what are the drugs that are inhibiting the pathway, and then what's the consequence of the drug in that pathway. So we've looked at benefit, and the benefit for placebo or treatment is the adenoma reduction. So one of the things, if you just look at the benefits going from the ODC GG genotype to the GA to the AA genotype over here, you can see the general reduction in overall risk of adenomas, just as we saw from the original Martinez study. The ODC A-allele appears to be prognostic for recurrence. The treatment works very well in terms of benefit in this GG genotype and the GA genotype, it doesn't work at all. In fact, may be contraindicated in this A genotype. And we think we understand the mechanism of why this happens. If you look at the risk here, and risk is now defined as the ototoxicity, there's absolutely no difference between the treatment and placebo in this genotype, in the heterozygous. Apparently, the majority of the risk is associated with this ODC A-allele, the minor allele, which has a very simple consequence, and that is don't treat that genotype in moving ahead. Now, the last point before I summarize is in terms of who are the populations that we should be targeting. We're talking about personalized prevention here. We are not advocating thinking about the general population. We are not thinking about treating people in the colon cancer area who have average risk of colon polyps, who have average risk of colon cancer. We're talking about really thinking about populations that are at advanced, have moderate to high risk. Why is that? This is a little table that I copied from John Potter's analysis of the APC and PRECEPT studies for nonsteroidal anti-inflammatory drugs that were published several years ago. And it just makes the point for 1,000 patients, what's the risk of death due to colon cancer or cardiovascular events, the total events, the toxicities of agents that increase this by only 2%. It's small, so this is 15 out of 1,000. If you doubled this, even if you reduce this by a half, the total number of events is not going to be changed in a positive way in this kind of a population. So one of the things that we're focusing on is let's look at higher risk populations. I've defined these down here: prior colon cancer, prior colon adenomas, even higher risk genetic factors. If you can now do things to maintain the negative effects of things like hearing loss, cardiovascular effects, then interventions begin to give you positive risk-benefit relationships. So in terms of our view of personalized prevention, we're thinking about starting with the general population and identify those people who are at higher risk: colon cancer survivors, prior adenoma formers, genetic factors, potentially inflammatory factors. The ODC A-allele is one of the genetic features, and we think there are going to be a number of other features that we can add to that that are going to provide us with don't treat or treat decisions. And I've included down here the exclusion of, for instance, 35% of people with high cardiovascular risk factors at baseline that are the numbers about that we and other people have associated with risk of nonsteroidal anti-inflammatory drugs. It identifies a significant population of patients that potentially could benefit from this kind of an intervention. What do you do with these populations? And then many of you are out there thinking, wait a minute, you're not going to be treating people with inflammatory bowel disease with nonsteroidal anti-inflammatory drugs. This gives opportunities for, again, predictive approaches using the ODC A-allele. They're the people who especially respond to aspirin. And we're thinking about non-steroidal anti-inflammatory drugs for this particular group of patients. So what's the overall effect that you might expect from this kind of an intervention? First of all, I need to make the point that I'm really emphasizing more that we think about what we're doing as treating risk factors, not preventing cancer. What we're really doing are treating those risk factors with the potential benefit that we're going to reduce cancers. If we start with these high-risk individuals here and then combine screening, combine lifestyle, diet, as I've talked with you earlier about, along with effective risk factor therapies, effective meaning potential to reduce the number of people with adenomas by 90%, has the potential thereby to reduce this 150,000 down to very significant numbers. This is a strategy that has the potential over the near future to remarkably reduce colorectal cancer deaths. And just to acknowledge a whole variety of people that have worked on this over the last number of years, colleagues at the University of Arizona, Frank Maskins, who's here in the audience, as I've mentioned, and colleagues at the University of California, Irvine, our clinical trials group, the National Cancer Institute, and especially Ernie Hawk, who's now at MD Anderson, and my company. Thank you very much.